Hello student, good morning. I am Dr. Ajay Kumar Sinha, Professor of Chemistry. Today I am going to discuss enzyme catalysis. Last class I have discussed adsorption theory of heterogeneous catalysis. Also I discussed shape selective catalysis by zeolite. Now I am going to discuss enzyme catalysis. Enzymes are complex nitrogenous organic compound. What is enzyme? Then enzymes are complex nitrogenous organic compounds which are produced by living plants and animals. They are actually protein molecules of high molecular mass and form colloidal solution in water. They are very elective catalysis. Catalyze numerous reactions, especially those connected with natural processes. Numerous reactions that occur in the bodies of animals and plants to maintain the life process are catalyzed by enzymes. The enzymes are thus termed as a biochemical catalyst, and the phenomena is known as biochemical catalysis. Many enzymes have been obtained in pure crystalline state from living cells. However, the first enzyme was synthesized in the laboratory in 1969. Some examples of enzyme catalyzed reactions I am telling you. First example, that is the inversion of cane sugar. Inversion of cane sugar. The inversion of cane sugar, here catalyst enzyme used. Actually, in a simple way, you can tell that enzyme is a biological catalyst. Enzyme is a biological catalyst. Very simple, in simple way. Then its example is inversion of cane sugar. This is example of enzyme catalyzed reaction. And here enzyme used is invertage. Invertage. In the last three letter is ASE. Invertage. According to the enzyme process name is given. Inversion of cane sugar is catalyzed by invertage enzyme. Okay. The invertage enzyme converts cane sugar into glucose and fructose. Then here cane sugar means C12H22O11. Aqueous solution of cane sugar plus H2O in presence of invertase ASE. Last three letter is ASE, invertase. It keeps glucose and fructose, means C6H12O6, and other fructose is also same formula. Both are isomer. Glucose and fructose are isomers, okay, having the same chemical formula. Second example I am telling you, conversion of glucose into ethyl alcohol. Conversion of glucose into ethyl alcohol. Here gymase enzyme is used. Here as catalyst zymase, ZYMASE. Zymase enzyme is used. Zymase enzyme converts glucose into ethyl alcohol and carbon dioxide. Means glucose C6H12O6, its aqueous solution in presence of zymase. It converts to ethyl alcohol and carbon dioxide gas. Actually, these two reaction actually is used in our body also. When we eat, then we eat a starch like rice, wheat item. When we are eating, then this starch is a complex molecules, bigger molecules. This starch converts to sucrose C12H22O11, and this sucrose it converts to glucose and fructose. Hydrolysis in presence of water, sucrose in presence of water, and catalyst means enzyme. Okay? Enzyme is invertase. I told you, in presence of biological catalyst, which we are calling enzyme, invertase, it gives glucose and fructose. And this glucose and fructose, both actually in presence of zymase, it converts to ethyl alcohol and carbon dioxide. Then this conversion of glucose into ethyl alcohol and carbon dioxide takes place in the presence of zymase, ZYMASE. Then this reaction takes place in our body. That's why after eating food, we feel sleepy due to the formation of ethyl alcohol, and we release carbon dioxide. Correct? Daily we are releasing carbon dioxide every minute. Then this carbon dioxide only. Other processes also there in our body that also releases carbon dioxide. But one process is this also. We are feeling sleepy due to the formation of ethyl alcohol. Ethyl alcohol means now wines. In wines, main composition is ethyl alcohol. Then here in a natural way it is forming in our body, and carbon dioxide we are releasing due to this process. Okay. Then two examples I have given you. Third example, conversion of starch into maltose. What I told, and that starch converts into sucrose. Then here sucrose means maltose. Both have same, you can say, chemical formula. Maltose and sucrose both have same chemical formula. Then conversion of starch into maltose, that happens in presence of enzyme. Its name is diastase enzyme. Diastase, DIASTASE, diastase enzyme. Okay, DIASTASE, diastase enzyme. This diastase enzymes converts starch to maltose. Okay, it converts starch into maltose. Maltose means sucrose. Then here starch formula you should know. This is actually C6H10O5 whole N. You don't know? There is a repetition of this molecule C6H10O5. This is a starch, but many molecules will be joined to each other. That's why whole N. It is represented by whole N. N means number n, maybe 10, 15, 12, any number, you don't know how many number. Then C6H10O5 whole n. Okay, then this is a starch. This starch reacts with water and it will give maltose. Maltose means sucrose, C12H22O11. Then this reaction takes place in a liquid, aqueous solution of a starch plus water. Means suppose you have taken 2 molecules of a starch, 2 molecules of a starch plus NH2O. 2 molecules of starch means 2 into C6H10O5 whole n, that is considered as a 1. N may be any value. Then 2 moles of a starch plus NH2O, because C6H10O5 whole n is there, that's why NH2O is required. Then it gives n moles of maltose. N moles of maltose means C12H22O11. Then here there is a role of diastase enzyme. Now next example I am telling you, conversion of maltose into glucose. Conversion of maltose into glucose. Maltose, you know, maltose actually that is a sucrose only. Already I have discussed cane sugar. Maltose is cane sugar, also called sucrose. Okay, total you can tell 2 other names are there, cane sugar or sucrose, same formula, C12H22O11. Okay, cane sugar, maltose and sucrose all have same chemical formula. All 3 have same chemical formula. Then this conversion of maltose into glucose means here C12H22O11, it's a aqueous solution, that is called maltose plus H2O. It gives directly, it will give glucose, C6H12O6. This is the glucose. Actually, cane sugar was giving glucose and fructose, correct? Here directly it will give 2 moles of glucose for the fructose and glucose, because formula is same, chemical formula is same, both are isomers. But here in this reaction, maltose on hydrolysis means maltose plus H2O in presence of maltase. Here enzyme name is maltase, ASE. It gives glucose. Okay. Now next example I am telling you, that is the decomposition of urea. Decomposition of urea into ammonia and carbon dioxide. Here which enzyme you will use? Then urease, UREASE, urease. Okay, here enzyme used is urease, UREASE. Okay, in the presence of urease, urea formula you know, CO will be in the middle, carbon monoxide, and both sides, left and right side, NH2. That is the urea. Then urea plus H2O, it gives ammonia gas plus carbon dioxide, means 2NH3 plus CO2 in presence of urease. Next example I am telling you, in stomach, the pepsin enzyme actually is present in our stomach. Don't forget this, whatever examples I have given, all are asked in the competitive examination. Okay, in every examination, even in UPSC also, this question is asked. Now 6th example I am telling you. Then all examples are these questions are asked in examination. Now I am telling you that in stomach, pepsin enzyme is found. Pepsin enzyme. Then in stomach, the pepsin enzyme converts protein into peptides. Protein into peptides. While in intestine, one is stomach and other intestine. Intestine, pancreatic trypsin. Pancreatic trypsin. This is also enzyme. Pancreatic trypsin converts protein into amino acids by hydrolysis. Here, protein actually this will be of high molecular mass. Protein that is actually made from amino acids. Amino acids having a very high molecular mass, that is called protein. Main constituent of protein is amino acid. Then this proteins actually convert into peptides in the presence of pepsin enzyme. Means pepsin plus water. This protein plus water, it gives peptide. And this pepsin is here enzyme. Similarly, in intestine, pancreatic trypsin, which is enzyme, converts protein into amino acid. Protein into amino acid, from which it formed, it will give back. Protein is formed from amino acids. Then protein on hydrolysis, means protein plus H2O, it gives amino acids by hydrolysis. And here enzyme used is pancreatic trypsin. Question is asked. One more example I want to give you, that is the conversion of milk into curd. Milk into curd. It is an enzymatic reaction. Here also enzyme is used. Okay, enzymatic reaction brought about by lactobacilli enzyme. This you remember. This in medical, many times this question is asked. All this question in medical, it is common. Always it is asked. Then lactobacilli, bacilli, 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 lactobacilli enzyme is used here. Means milk in presence of bacilli enzyme, it converts into curd. Okay, that's why if you want to convert milk into curd, then a small quantity of curd, you are after heating the milk, you have to cool it, and when it is in the little warm, that time you have to mix a little curd, because in this curd that enzyme will be present. Okay, and then milk will convert into curds. A small quantity of curds we are using, because this catalyst is used. Enzyme is present in that catalyst. Okay, lactobacillus, mainly that enzyme will be present. That's why because you will not get direct bacillic enzyme, lactobacilli, lacto means milk actually, lactobacilli is present in curd. That's why directly a small quantity of curd you are mixing in warm milk, and then it will convert to curd. Now another characteristics I am telling you, some characteristics of enzyme catalysis. What are the this feature or characteristics of enzyme catalysis? I will give some characteristics. I will tell about some features. Then here enzyme catalysis is unique in its efficiency and high degree of specificity. Enzyme catalysis is, I told you, unique in efficiency and high degree of specificity. Then first characteristic I am telling you that most highly efficient. First characteristics, highly efficient, most highly efficient. One molecule of an enzyme may transform 1 million molecules of the reactant per minute. Just you see that it's a highly, you can say, efficient property. Most highly efficient, that one molecule of an enzyme may transform 1 million molecules. 1 million molecules of the reactant per minute. Now, second characteristic, highly specific nature. Highly specific nature, I am talking about the enzyme characteristics. Then highly specific nature. Each enzyme is specific for a given reaction. That is, one catalyst cannot catalyze more than one reaction. For example, the enzyme urease catalyzes the hydrolysis of urea only. It does not catalyze hydrolysis of any other amide. Okay, one enzyme can hydrolyze only one reaction. This is the characteristics. Now, third that is the highly active under optimum temperature. Means it requires a specific or fixed temperature. Only that time it will react very fast. Highly active under optimum temperature means required temperature. If temperature is more or less than this optimum temperature, then it will not react. Okay. The rate of an enzyme reaction becomes maximum at a definite temperature called the optimum temperature. On either side of optimum temperature, means less or more, enzyme activity decreases very less. Okay, maximum will be at that optimum temperature. The optimum temperature range for enzyme activity is 298 to 310 Kelvin. 298 means 273 plus 25 means 25 degree to 35 degree. Okay, 298 to 310. Then 298 means what? 298 means 25 degree plus 25 degree, and that is 298 plus 37 you can tell. Okay, 37 degree. 298, between 298 to 310 Kelvin. Human body temperature. Our body temperature, how much? Human body temperature being 310 K. That is the 37. Okay. Then K is, this temperature is our body temperature, is suited for enzyme catalyzed reaction. That's why in our body so many enzyme catalyzed reaction takes place. Okay. Now, next characteristics, highly active under optimum pH. pH, what is the pH? pH is the negative logarithm of hydrogen ion concentration. pH is equal to negative logarithm base 10. negative logarithm of hydrogen ion concentration. It depends upon hydrogen ion concentration, means H plus. Okay, and you know that pH value for neutral, that is the neutral water, is 7, and below 7 is acidic. Below 7 is acidic, and after 7 is basic. Okay, then this is the pH value. Okay, pH means less than 7. pH scale you are knowing. Then less than 7 is acidic, more than 7 is basic. Then here it also requires fixed pH value. Fixed pH value, that is between 5 to 7. pH value must be between 5 to 7, means little acidic. Okay, the rate of an enzyme reaction, enzyme catalyzed reaction, is maximum at a particular pH called optimum pH, which is between pH values between 5 and 7, maybe 5, maybe 6, maybe 7. Next characteristics, next feature, increasing activity in presence of activators and coenzymes. Okay, means activity, increasing activity in presence of activators and coenzymes. Means a main enzyme is present. With enzyme, other activators who will enhance the, who will support the enzyme, that is called activator, who will keep activated into enzyme, and also coenzyme. These two, activator and coenzyme, increases the activity of enzyme. The enzymatic activity is increased in the presence of certain substances known as coenzymes. It has been observed that when a small non-protein means vitamin, is present along with an enzyme, the catalytic activity is enhanced considerably. Okay, activator are generally metals. Metal ion may be there. Activator means metal ion. Okay, and this I told you, vitamins, non-protein, it behaves as a this activators, and not activators, coenzyme, that is the vitamins, which is non-protein, behaves as coenzyme, and activator is a metal or metal ion. Activators are generally metal ions such as sodium ion, manganese ion, Na plus, Mn2 plus, cobalt 2 plus, copper 2 plus, etc. These metals ions, when weakly bonded to enzyme molecules, increases their catalytic activity. Amylase, in presence of sodium chloride, amylase is AMYLASE, amylase, it is enzyme, catalyst you can say, enzyme, in presence of sodium chloride. Sodium chloride produces sodium ion, are catalyzed very, it will enhance, you can say, catalytically, means it will to protein in presence of amylase, or amylase in presence of sodium chloride, that is the sodium ions are catalytically very active. One more characteristics I want to tell you. Influence of inhibitors, inhibitors, and poisons, both functions negatively. Okay, Influence of inhibitors and poisons. This question is asked. Always this question is asked. Okay, all questions are important. All questions are asked sometimes in all examination, in general competition also. Like ordinary catalyst, enzyme are also inhibited. Inhibited means made slow or poisoned, can stop completely also by the presence of certain substances. The inhibitor or poisons interact with the active functional group where it will attack. These inhibitor or poisons, it will attack or it will interact with the active functional group on the enzyme surface and often reduce or completely destroy the catalytic activity of the enzyme. The use of many drugs, example I am telling you, drugs are used, okay, the use of many drugs is related to their action on enzyme inhibitor, as enzyme inhibitor in the body. Okay, this drugs, many drugs, suppose something happened, some reactions are going on, you want to, you can say, reduce the effect or stop that reaction, then some specific drugs are used. Because sometimes you are taking medicine and it is suppose wrong medicine you have taken, then you have to stop their reaction. You have to stop their reaction. Suppose anybody has taken poison, then now you have to cancel the effect of poison or wrong medicine or anything, something is very reactive things he has taken, then to stop that reaction some special drugs are used. Okay, and that drugs will interact with the enzymes. Okay, and reaction will stop. Drugs is related to their action as enzyme inhibitor in the body. This I have told you, these things, feature or characteristics of enzyme catalyst, okay, or enzyme catalysis. Now I am going to tell mechanism of enzyme catalysis. Mechanism, how does it work? Already I have given little idea. Now I will tell you little details. But here one example I will tell you, then quickly you will understand. Okay, diagram also you can understand. Now I am telling that things. Actually, this mechanism of enzyme catalysis. Here actually how will it work? Both enzyme and substrate, enzyme or reactant, enzyme and reactant, how both will react, where it will attack, that point is very very important. Okay, I am telling you just simple example. First, suppose you want to put fire on wood, then where you will apply this fire on wood, where you will apply, then you will apply fire on the corner of wood. Suppose cube, you have taken a cube, then pointed, where pointed point is there if you will put the fire, then it will catch fire only that point. Or any wood where pointeds are there it will catch fire soon. Okay, you have seen also all the things. Then why on the pointed, why they are catching fire on the pointed that spot, point spot. Why? Because that where point is there or very thin is there, very thin. There actually at that point molecules or atoms will be there. Molecules or atoms upper part will be there. Inside wood, upper side only it catches the fire, because upper portion atom or molecules will be there, and atom and molecules whatever be, it will be very reactive and active. That atom or molecules will be excited soon in comparison to the middle. Okay, that's why pointed side now atom will be there. Atom will be there in the corner, then it can be excited soon, and when it will be excited, it will catch fire. Okay, and when it will catch fire, then that fire will give to the neighboring atom or molecules, that will be also excited. Again same thing will happen, it will again give further neighbors. Like that way all wood will burn. Then there is a spot, fixed spot, where atoms and molecules will be in a position to react. Only that position, you can say other reactant, suppose both reactant are there, then both will attack only, first they will attack only that type of you can say point or a spot where they are in atomic form or molecular form. Then here also mechanism of enzyme catalysis. Here also just simply I am telling you, just like a lock is there, lock you are opening with key, correct? Other places if you this key you will enter, then it will not open. Right place, key and lock. Keys must be go in lock, and in lock also we are entering that hole, it should be in a proper position, otherwise it will not open. Then like that situation is here, enzyme will attack the substrate or reactant on that type of spot. Okay, that's why it is also called these things lock and key. Okay, this is also called a lock and key mechanism. Now I am going in just in details. And there are you can say, and there are a number of cavities present on the surface of colloidal particles. Okay, here actually enzyme that will in the colloidal form. Then there are a number of cavities present on the surface of colloidal particles of enzyme. They will be in the particle form. Enzymes will be in the particle forms. These cavities are of characteristics shape. These cavities means small holes will be there. Then these cavities are of characteristic shape. Its shape is fixed and possesses active group. These cavities possesses active group such as NH2 group, carboxyl group means COOH group, SH group, OH group, etc. Then these are the groups. You know that these groups takes part in the reaction. These groups takes part in the reaction. Wherever these groups are there, only there reaction will take place. Then in cavities, these groups are present. That's why substrate or reactant will attack on that cavities. Okay, these are actually means I am talking about groups, present in the cavities. Then these are actually the active center, you can tell, active center on the surface of enzyme particles. The molecules of the reactant, also called substrate, which have a complementary shape. See, this complementary shape means here that I told you in a lock key will go. Then key and lock both have complementary shape. And lock, that hole will be there and key will be there. Then key will fit in that hole. Key will fit in that hole. That is called complementary shape. Means it will set there. Key will set inside. Completely will set. That's why it is called complementary shape. And it will fit into this cavity. Then this substrate or reactant, it will fit in the cavity of enzyme, just like a key fits into a lock. On account of the presence of active groups, an activated complex is formed, which then decompose to yield the products. After this, when substrate will react with enzyme, then when both are fitting, means key is fitting in lock, correct? Then that situation is called activated complex. Means enzyme plus products, now both are reacting, both are attacking to each other, reacting. Then they will form one product complex. They will form an active complex, or also you can tell enzyme substrate complex. It forms an activated complex, and this activated complex formed, which then decomposes to yield the product. Correct? Actually, this enzyme, when it attacks the substrate or reactant, it will divide it into two parts. Correct? That's why I already told you that HI, suppose decomposition of HI, then H and I, now molecule can break into H and I. 2HI will react, HI plus HI, then they will form HH and II, means H2 and I2. Then at activated position, complex, 4 bonds finally it will be there. 2 H and I old bond, again another H and I old bond, 2 bond, a new bond will be formed at intermediate stage or activated complex. That time transition state, what we are telling, there old bond will begin to break and new bond will begin to form. Hence all four bonds will be present at transition state, and slowly as the strong bond will be formed, weak bond will break completely and it will form H2 plus I2. Then here same type of things are there. Here you have taken both enzyme catalyst plus substrate, means reactants, and both will react at active site, and it will give enzyme substrate complex. It will give, both are interacting, and this enzyme substrate complex, it will be separated. Enzyme will be separated because it is a catalyst. You know, catalyst does not take part in the reaction. Then enzyme will be separated and product will be separated. Okay. Then this I told you, mechanism of enzyme catalyzed reaction. Okay. Then here enzyme catalyzed reaction may be considered to proceed in two steps. First step, suppose E, capital E, means binding. First step is binding of enzyme to substrate to form a activated complex. Means E plus S. Capital E means enzyme plus S. S means substrate. Then they will form bond, weak bond, ES. Complex is known as ES, and one star you can write. This is the active. To mention, to show the activity, a star sign is added. ES, one star. Upside you can write a small star. This is called activated complex. This is the first step. And second step, decomposition of this complex. Decomposition of activated complex to form product means ES star. ES star will break into what? E plus P means product. Activated complex will give it E. Enzyme it will return, and product it will form P. Then this ES star, that is the complex, will give enzyme plus product means E plus P. This I have discussed. Okay, mechanism. Now I am telling you some I can say catalyst in industry. Which catalyst they are using in industry, purposeful work. Actually, our main intention where catalyst is going to be used, correct? And what you will get, where it is used. Application of catalyst also you can tell. Then its main use is in industry, in certain reactions, and that reaction gives useful product. Okay, then I am giving some examples. Then here first you can say process is Haber process. Haber process for the manufacture of ammonia gas. Then N2 plus 3H2. N2 plus 3H2, it gives 2NH3. Here which catalyst enzyme you will use? Which enzyme? Enzyme you consider as a biological catalyst. Don't forget, in a easy way. Okay. Then in this reaction N2 plus 3H2, it is giving 2NH3 ammonia gas. Then which enzymes are used here? Catalyst. Then finely divided iron. And molybdenum is used as finely divided iron. Molybdenum as a promoter it is used. Okay. And what are the conditions? Means two things you are using. Iron is a catalyst, molybdenum is a promoter. Question is asked. Okay, in all examination. And what are the conditions? Then pressure should be 200 bar. 200 bar pressure, and temperature, that is the 723 to 773 Kelvin. 723, between 723 to 773 Kelvin. This temperature is required. Nowadays a mixture of iron oxide, potassium oxide and alumina is used. This question is also asked. Nowadays mixture of three means iron oxide, FeO or Fe2O3 you can tell. Potassium oxide means K2O, alumina Al2O3. These three, mixture of these three are used. Mixture of iron oxide plus potassium oxide plus alumina is used here in the Haber process. Nowadays question is asked. Now second example I am telling, second process, that is the Oswald process for the manufacture of nitric acid. nitric acid is manufactured by Oswald process. This question is also asked. Then here reaction is 4NH3 plus 5O2. 5O2. It gives 4NO, nitric oxide, plus water, 6H2O. Means 4NH3 plus 5O2. It gives 4NO plus 6H2O. And this NO, further it will react with oxygen. 2NO plus O2, it gives 2NO2. And in the third reaction, this 2NO2, further it will react NO2 plus water, means 4NO2 plus 2H2O. And oxygen. In every step reaction is used. Okay, means 4NH3 plus 5O2, it gives 4NO plus 6H2O. And this NO, 4NO means NO, that will react with oxygen, means 2NO plus O2, 2NO2. And this NO2 will react with oxygen in water. Oxygen in presence of water, 4NO2 plus 2H2O plus O2. It gives 4HNO3. Now you are getting nitric acid. Then here which enzymes you are used or catalyst you are using? Then platinized asbestos. Platinized asbestos. On asbestos sheet, that platinum layer will be there. That is called platinized asbestos. Temperature, how much temperature is required? Then 573 K Kelvin. Now one more example I am telling you. In contact process. In contact process for the manufacture of sulfuric acid. This question is also asked in all examination. Okay. Whatever I am telling you, highlighted question, you keep in mind from the chapter. Each chapter I am telling this will be asked, that will be asked. It means that questions are important. And it is not fixed for that it will be asked only in main or advanced or in medical. Anywhere, they can change the little question, they will twist it. But can be asked in any examination, even for general examination also. Then now in contact process, by this process, contact process, sulfuric acid is formed. Then here also 3 steps are there. Sulfuric acid is formed in 3 steps. First, sulfur dioxide, oxidation of sulfur dioxide means 2SO2 plus O2. It forms SO3. And SO3 now formed, it will react with a concentrated H2SO4, little amount of I can say H2SO4. Then it will give H2S2O7. This is called oleum. Oleum, O-L-E-U-M, oleum, H2S2O7. Okay, in this process you are using little concentrated aqueous H2SO4 you are using here. Then first step, SO2 plus O2, it gives SO3. Second step, SO3 plus dilute H2SO4. Dilute means aqueous, you can tell. It will give H2S2O7, liquid. Oleum is the liquid. Now third step, this H2S2O7 will react with water. H2S2O7, which is liquid, means oleum plus water, it will give H2SO4, 2H2SO4. Okay, that is the sulfuric acid. Now concentrated, you got concentrated sulfuric acid, but little amount of sulfuric acid you have used. Aqueous H2SO4 you have used in the second step. Then here, what you will use as a catalyst? Then here again, platinized asbestos you will use. Platinized asbestos, as in what you have used in nitric acid here also, but here in H2SO4 a vanadium pentoxide is also used. And extra main things you can tell, vanadium pentoxide V2O5 is also used here. Okay, question is asked, either platinized asbestos or vanadium pentoxide, and temperature 673 to 723 Kelvin. 673, between 673 to 723 Kelvin. Okay, and this I have discussed about the this things, enzyme, mechanism of enzyme catalyst, and catalyst used in industry I have discussed. Okay, now next class I will start colloids. Okay, colloids I will start in next class. Thank you.